Good morning, everyone, and welcome to today's MPG Primer, the first of the 2022 to 23 series. We are grateful to have two speakers here today from the Marks Lab. Dr. Nikki Thadani, as a graduate student, worked at Rice University, but since then has come to the Broad, where she has worked on viral evolution and data-driven protein engineering as a postdoc in the Marks Lab at HMS. Her previous work focused on synthetic biology to engineer viruses for gene therapy. Presenting with her is Sarah Gurev, who's a graduate student in the Marks Lab from MIT EECS. She previously worked in structural biology as an undergraduate at Stanford. They'll be presenting their work today on learning from pre-pandemic data to forecast viral antibody escape. They'll be happy to take your questions at the end of their talk, so go ahead and type in the Q&A and we'll ask them in the order in which they appear. Or at the end of the talk, you can raise your hand and voice your question. So thank you both for being here today. We look forward to your talk.
Great, thank you so much for having us. We're really excited to be here and tell you a little bit about what we've been doing. And just generally, we're really excited to have some discussion at the end. This is an ongoing project for us, so we have the preprint kind of on the first phase of the work, and then we'll be talking a little bit about the ongoing phase as well. So when we came into this space thinking about viral mutation, so this is a plot showing the course of the number of mutations we've seen in SARS-CoV-2 over the pandemic. We came into this space thinking about what do we need to know to make some actionable interventions for this pandemic. And so we kind of came into that with our central goal of being able to provide information that could be useful for intervention. So there are a couple different types of interventions you can think about. I think one of the most common ways to think about this, and what really drew us into the space, was the idea of what could we say about which mutations might escape immunity to inform the development of vaccines. So when we think about when we might want to know this, we do need a pretty decent lead time for this, because the development of a vaccine takes time. Even now that we have a vaccine going and we're just updating the strains, the approval process and selecting which strain to include takes several months after that strain first appeared. For example, with Omicron, we're just starting to see deployment of vaccines, including that strain now, and its original appearance was in late 2021, so still a pretty decent lead time on that. But even once you are deploying the vaccine, you don't want that vaccine to immediately become useless, right? You would hope that you'd be able to give that vaccine out, then ideally, hopefully people can have immunity for at least a year. And so we think of this, we'd like to be able to say something about which mutations might be relevant for escape roughly 2 years out. Another thing that we might be able to help with is understanding which antibodies might be most useful for monoclonal therapeutics. So for this, I'm using 18 months as a proxy for what I've heard is typically a good number for the development of a monoclonal antibody as a therapeutic. And there's kind of another end of this once you're getting a little bit further along. We think of this, the next 2 points, as more of in the forecasting realm. So kind of trying to say in the midterm to near term, what do we expect to see? We would like to be able to say something about which mutations are going to arise on top of the strains that we're currently seeing and which are currently prevalent. And so we think about that as being able to understand the epistatic constraints of the new strains that we're seeing, and then say, okay, what is the most likely mutations we're going to see on top of BA4, BA5, things like that, the current Omicron concerning strains. And then on a shorter time scale, it's also useful to understand which strains are most likely to become dominant. So of the ones we're seeing now, which ones do we expect to kind of take predominance in the pandemic? And these last two things are both useful, kind of less for the development of interventions and more for their strategic deployment. So if we know, for example, that a strain that is resistant to one of the current monoclonal therapeutics is taking over in India, that therapeutic might not be deployed as much in that region, right? So we can kind of adjust our response to the pandemic using this type of information. So this is just to kind of frame the general goals of prediction. Our talk will mostly focus, or we'll start out focusing on the early sites, and that's kind of what motivated us originally. But towards the end of the talk, Sarah will talk a little bit more about the second half of this as well, which is what we're working towards currently with some cool collaborations. So with that motivation of trying to make some statement about what's going to happen in the pandemic early on, we start thinking about what a perfect predictor would be able to do. So first, we want our predictor to be able to say something early on. We're really interested in being able to make predictions in the beginning of a pandemic. So just when we first start hearing about a pandemic virus, the consequence of that is we're unlikely to get a lot of information about that pandemic, both in terms of experiments to really probe how that virus works, as well as sequencing data early enough for us to be able to make the sort of predictions we'd like to make. As the pandemic progresses, we do want to be extensible to variants. So like I mentioned on the previous slide, the idea of as strains appear, we want to be able to say whether our predictions are still valid for those backgrounds and update our predictions for those new backgrounds. For example, with SARS-CoV-2, the D614G variant took dominance in the pandemic fairly early compared to other mutations. And it would be useful to have a model which can quickly say, okay, now that we're seeing this mutation, how does that affect our predictions? We want our model to be adaptable to new data. So we want to be able to update how our model works as we do have pandemic information available. Once we start to get more experiments through the pandemic, or once we start to get a wealth of sequencing data, we wanted a framework that we could adjust. And finally, we were hoping to develop a model that is relatively biologically interpretable. So when we say that we think a mutation will be able to escape antibodies, we'd like to be able to say something about whether we think that means it's fully just displacing antibodies, or whether it's increasing viral fitness in some other way to compensate for antibody targeting, for example, stronger receptor binding or things like that. And so these were kind of the goals we set for ourselves. We also went into this kind of with a structured biological framework of what we think an escape mutant should be able to do. So when we think about the likelihood that a mutation will escape prior immunity, we expect that that mutation needs to retain enough of viral function for that variant to continue to be viable as a pathogen. So that means that variant needs to still fold and form a virion correctly. It needs to interact with host receptors, and it needs to be able to transmit between hosts. We expect for a mutation to displace an antibody, that mutation also needs to occur in or near where an antibody binds the protein. And so it needs to be, that mutation needs to occur somewhere that is accessible to antibodies, or at least close to regions that are accessible to antibodies. And finally, for a mutation to affect antibody immunity, we would expect that that mutation needs to interrupt that interaction between the antibody and the protein. So when we think about antibody escape, we're thinking about antibodies that have been previously risen against this target, either from vaccination or prior infection. And then we're looking for mutations that are changing the surface of the protein enough that they displace those interactions. So this is kind of the framework we went into this. So with this, we started thinking about how we could start to kind of make these predictions with very limited data from the pandemic. So what we could say about which mutations are likely to escape with the very initial information that would be available with the SARS-2 protein sequence and maybe some initial structures. In one area, we decided to look for information about protein evolution is by looking at deeper evolution. So in this case, going deeper into coronaviral evolution beyond just the SARS-CoV-2 variation. This is an area that our lab has been interested in for a while and has leveraged for a couple different areas. So it was a natural place for us to look. So here what I'm showing is basically a 2D representation of sequence space. Here we're deriving it from the latent space of an autoencoder. But you can also look at visualizations like this from PCA or other ways to create them, just getting a sense of the spread of coronavirus family sequences. And what's shown in green is what we're referring to as deeper evolution, so the broader coronavirus family beyond the SARS variants. And then in the center, what I've boxed is the variation that we see for SARS-CoV-1, so the original SARS pandemic virus, and then SARS-2. Excuse me. So the main thing that is notable here is when we think about this as the spread of sequence distances, there's a much larger spread of diversity in the broader evolution than what we see in pandemic evolution. So in addition to this data being available early, we expect that we would have broader viral evolution sequences available at the beginning of a pandemic. This extensive variation can also be useful for predicting future mutations that haven't yet been seen in the course of a pandemic, but might have been seen across broader evolution. And the nice thing about this broader diversity is it's also useful for informing models that can learn about the constraints between positions in a protein alignment as opposed to just single sites. So we're more likely to be able to learn epistatic constraints on the protein. So one example of our lab using this previously with quite a bit of success is the evolutionary model of variant effects. So here, the way we chose to use this data, or the way others in our lab developed to use this data, was that they used a variational autoencoder trained on an alignment of deeper protein evolution for a given human protein. So they built an alignment of protein variation across the tree of life. And then once they had that alignment, they trained this autoencoder, which essentially is trained to reproduce the sequences that it is trained on. So that's kind of the loss function it's trained on. And it compresses information from those sequences into a restricted, smaller latent space. So it learns the most important information from those sequences to be able to reconstruct the distribution. So once they trained this model, then they applied it to make predictions about human genetic variation. So they looked at a protein. Here's an example of P53, and they made predictions of all the single residue substitutions in the protein from this model, and they saw how those predictions corresponded to human pathogenicity as reported in ClinVar. And they find that these predictions, which are derived from broader evolution, so from across the tree of life, are actually really powerful for making predictions for human genetic variation. And in particular, this type of approach is really exciting for being leveraged for variants of unknown significance. So if you identify a variant and you have no idea whether it's a likely contributor to a patient's disease, this model can provide some insight for variants that haven't been widely characterized in humans yet. And so this work was actually presented at MPG, I think, a year ago, but beyond that, by Johnny and Mafalda from our group. This is their paper. I don't think their MPG talk was posted online, but I've also linked their MIA talk at Broad. So definitely check that out if you want a refresher on that. Really exciting work, but we were kind of inspired by this and by the framework of their model to apply a similar framework to viruses. So what we did was we took kind of a similar concept, but using the broader viral evolution that I mentioned previously. So we trained the similar variational autoencoder framework using broader viral evolution. Here we're not using pandemic sequences. So we explicitly dropped out anything that was seen other than the original first Wuhan strain that was sequenced. We're not including any more pandemic variation. Other than that, it's all pre-pandemic sequences, and we trained our model on this. And then we decided to look and benchmark and see whether our model is capturing features that seem to be predictive of what is required for SARS-CoV-2 to function. So to do that, we made predictions for the spike RBD, the receptor binding domain, and we compared that to deep mutational scans that exist for this domain. So the Jesse Bloom lab and a few others have done really extensive DMS studies by displaying this RBD domain on the surface of yeast and then evaluating whether it expresses, whether it binds to ACE2, and as I'll get into later, also whether it escapes immunity. But here, when we compare our model predictions to the expression ACE2 binding experiments, what we find is that the sites or the mutations that successfully both express and retain their binding to ACE2 are the ones that have high scores under the model. And so this was encouraging to us because it suggests that the model is capturing some of these important constraints that might be necessary for spike function, right? We imagine that the RBDs that are expressing successfully on yeast are at least retaining their fold and able to fold successfully. And then of course they need to bind ACE2 in order to be infective. So while that was promising for the question of fitness, we were a little more hesitant about using this type of deeper evolution for immune constraints. One reason for this is it's known that immune selection pressures are quite different across different host organisms. For example, even comparing bats to humans, bats rely more on innate and T cell responses than their B cell responses. And their B cell responses are also quite different in a number of ways. So we would expect that the types of constraints we're seeing across all these different animals that are hosts for coronaviruses that are shown here might not be super predictive of what we care about, which is how which mutations will escape the human B cell response. And of course, in order to get the specific human response information in a pandemic, we would need a large swath of the population to be immune, to either have been vaccinated or been infected. And we wouldn't want to wait for that to happen for us to be able to make at least our initial predictions. And so instead, we built a model framework, assuming that we wouldn't have that data available. So our model framework using pre-pandemic data is to incorporate EVE as our fitness component, because we've shown that that is promising for this type of application. And then for accessibility, we just started with a simple assumption using the protein structure, so just the protein structure of the target protein that we're evaluating. In this case, we used a structure of SARS-CoV-2 spike. You could imagine that this requires a structure to have been resolved, but without a resolved structure, if you have a homologous viral protein, the structures are actually fairly comparable. So you could use a previous homolog and also use a predicted structure as well. But in this case, we use the SARS-CoV-2 spike protein structure. And then we just made an assumption about approximating from the structure which regions of the protein are most likely to be accessible. So surface accessibility, and also incorporating some information about the distance from the center of the virion. Then we also looked for this dissimilarity constraint just at single residue properties. So we looked at the difference between the starting residue and then the new mutation residue in terms of chemical properties, just hydrophobicity and charge metrics. And so with this simple framework, using entirely pre-pandemic data, we're able to make at least some predictions about which mutations we think are going to have the highest escape potential. So next we thought about, now that we have this framework, how do we evaluate whether it works? And when we think about this, we're thinking about it as a temporal experiment, basically going back in time and saying, if we were to make these predictions in the first few months of the pandemic, how would they have looked given what we know now? So now that we have more information about the pandemic, how would these predictions have looked? And the types of information we have a couple different types that are both useful and flawed, right? So there are a number of different experiments that we can compare to that are now available. There are the deep mutational scans that I mentioned, which have more high throughput information. And then there are also more low throughput, but probably closer to the desired phenotype experiments, looking at individual strain neutralization and things like that. These assays are able to really pinpoint the key functions we're interested in related to escape. But of course, all these assays, and particularly high throughput assays, can be an imperfect proxy for the full function that we care about. So for example, the RBD DMS that we're looking at, they are just focusing on the RBD on its own. These experiments won't capture any constraints that mutations to the RBD have in terms of affecting the overall spike structure. And so we would miss a little bit of information with those types of experiments. Another source of information we have through this pandemic is surveillance sequencing. So one really interesting thing about the SARS-CoV-2 pandemic as opposed to earlier pandemics is the huge wealth of sequencing data that has been gathered throughout the course of the pandemic. And this information provides somewhat of a comprehensive picture about how the virus is responding to all the constraints that it's subject to and which viral variants are succeeding under those constraints. However, there are, of course, other factors that go into which sequences take predominance beyond just their sequence. There's epidemiological factors that play into this. For example, whether a certain new variant arises in a region where there's a lot of transmission and a dense population versus a more sparse population. And also this data has various biases. We sequence certain regions of the planet a lot more extensively than others. And so we're more likely to get information about certain regions than others. So all this to say that these are great sources of data that we can compare our predictions to, but we're kind of working in a space where there is no ground truth. And so all our evaluations are subject to some caveats. I think a perfect ground truth was if we could get sera samples from every human on Earth and get a general average of how their sera responds, and then test variance against that. But we're trying to kind of get as close to that as we can with the data that we have when we're making our comparisons. So we started out with comparing to the deep mutational scanning experiments that I mentioned previously. And the way these experiments are conducted is using using a yeast-displayed RBD library and evaluating the ability of these variants to bind to antibody or sera samples from the pandemic. And so these experiments have been conducted by the Jesse Bloom lab and the Sunny Shi lab, and they're up to almost 400 samples of antibodies and sera. So when we make our comparison of our predictions to these experiments, we're particularly interested in how good is the top end of our predictions. The reason we wanted to zoom in on this is because we imagine if our predictions were to be used to develop a vaccine, you would take the top X number of sites that we say will escape by our ranking, and then those would move into initial experimental characterization, and potentially some of those mutations could be incorporated in a multivalent vaccine. So the first thing we wanted to ask is how accurate is the top, for example, 10% of our predictions. And when we compare to this data, we find that 60% of our top 10% predictions are observed to reduce antibody binding in this deep mutational scan. So this is exciting because we're at least capturing a good number of our predictions do reflect something that's been seen in this deep mutational scan. We also looked at kind of the other question, which is how many of the escape mutations that happened in the deep mutational scan do we capture in this top 10%? And that's about 35% of them. So in our top 10%, we're capturing 35% of the total number of escape mutations observed across these experiments. And when we compare this performance to the components of our model, so particularly zooming in on the fitness component, EVE, and the accessibility component, we find that our combined model does better at both of these metrics, so both precision and recall. And when we compare to another published predictor of escape, so this is the He et al. Paper that came out in Science in 2021, where they used a language-based model to try and derive predictions of which mutations facilitate escape. Our framework does a little bit better at capturing which mutations are most likely to escape. And so when comparing to the DMS, this was encouraging about our performance. We next dug in a little bit deeper to look at exactly where we are capturing escape mutants and also where we might be getting things wrong. So first, we just took a quick look at our predictions as they look on the structure as compared to where observed DMS escape has happened, and also known antibody footprints. So we looked beyond just the DMS data, and we looked at all the antibodies that have been reported in crystal structures with the RBD in the PDB, and we looked at their binding footprints as well. And what we see is that our predicted escape mutations line up pretty well with what has been seen in the data in terms of both the DMS data and then where antibodies bind. A lot of this is focused on the interface between the RBD and ACE2, so it's target receptor, which makes sense because those antibodies that interfere with that interaction between the virus and its receptor are most likely to be neutralizing and completely block infection. We also zoomed in on this a little bit and focused particularly on mutations or sites that we predict to have escape mutations, but which did not have escape observed in the DMS. And what we see is the vast majority of these sites actually are in the binding footprint of a known RBD antibody. So 88% of these predicted escape mutants that were not observed as escape in the DMS are in the binding footprint of a known antibody. So this could mean that since the DMS, while it has looked at a good number of antibodies, it obviously hasn't looked at everything. It could mean that as the DMS continues to incorporate more antibodies, we'll be able to see escape at these sites as well. Another thing we looked at was we kind of subset the data from these DMSs to zoom in particularly on the polyclonal immunity samples that they have, so the patient sera. These are sera samples from patients that either have had a prior SARS-CoV-2 infection or that have been vaccinated. And so we were interested in zooming in on this because this is kind of the closest proxy to the ideal data that I mentioned of getting a number of patient samples to get a full picture of their polyclonal immunity. This is not that many samples, and so we didn't use it as our only analysis, but we did want to see how we did on these particular samples. And what we find is that more than 50% of our top 10% predictions are sites of known sera escape. And so it does seem that our approach is capturing these mutations that from this data we think are the most relevant in terms of pandemic immunity.
Now, moving on from comparison to experimental data, we'd like to look retrospectively how our model is able to anticipate mutations that have occurred throughout the pandemic. We look at the wealth of sequencing data that is in GISAID. At this point, there are more than 11 million sequences deposited, where almost 99% of sites in spike have been mutated. We can see here the increase in variation in spike over time as we've gone through the different waves of the pandemic, first alpha and then data, and then later Omicron shown at the bottom, and the corresponding increase in variation in spike as a result. Moreover, we can also look specifically at variants of concern. These are not derived just from sequencing data, but actually as a result of assessment by the World Health Organization. They define VOCs by looking at sets of mutations that change the virus significantly enough in order to pose a global public health risk. This means genetic changes that impact viral characteristics like fitness, through changes in transmissibility or epidemiology, as well as viral escape, which would mean a decrease in the effectiveness of vaccines or therapeutics. It can take some time for variants of concern to be identified from the first occurrence, often 1 to 2 months. Though in the case of Omicron, where there was a significant number of mutations compared to what had been seen at the time, this was much faster. When comparing EVEscape scores to the pandemic, we don't necessarily expect that EVEscape would be able to capture all mutations that have occurred, but rather perhaps capturing mutations that cause escape rather than fitness, and perhaps not seeing those mutations that might have just been carried along without directly impacting either of these. So while we don't evaluate against just say and expect to see all these mutations, it's encouraging to see trends. We do see on this plot on the left that EVEscape is able to capture frequent pandemic mutations, shown in the structure as these more yellow mutations that are present in the spheres, which are those sites where mutations have occurred more than 10,000 times. We can also see that high EVEscape scores are more concentrated in immunodominant regions, such as the ACE2 binding region of the RBD, which is zoomed in on the structure, as well as the NTD on the left. We can also see this trend of high EVEscape scores relating to pandemic prevalence by seeing the high frequency of mutations that have occurred through the pandemic in the top decile of EVEscape performance. We see also that EVEscape scores may better anticipate variation as new selective pressures occur throughout the pandemic. So early in the pandemic, neutral drift is likely more relevant than positive selection. This can be seen through experiments, including DNDS. DNDS is this ratio of non-synonymous divergence over synonymous divergence, where a score of less than 1 means there's likely more neutral selection, where later we'll have more positive selection with a DNDS score of 1. So for spike specifically, we can see around November 2020, this shift to positive selection. This is around the time of the alpha VOC, and this is when there's still many naive hosts in the population. So when there's many naive hosts available, there'll be limited adaptive immune pressure, but as there's more vaccinated or previously infected population, there'll be more escape selection, immune selection later on. So we can see here on the right are trends of EVEscape as related to variation in the pandemic. We break EVEscape scores into 4 quartiles and see to what extent each quartile captures spike mutations that have occurred and seen more than 1,000 times in GISAID. So at that point where positive selection is starting to occur, that's where we kind of see a break off between the upper quartile and the lower quartile EVEscape scores. And Then later on, we can see there's this divergence between these two quartiles around 7%, which then even increases later on to 12%. So here we could see that EVEscape scores are continuing to better reflect the pandemic as the pandemic has progressed. Moreover, we see that EVEscape particularly captures frequent variants of concern mutations. A third of these frequent mutations are the top 10% of our spike predictions. We can see that here when looking at a plot comparing EVEscape scores to EVE scores, where EVE is just the model for fitness. We can see when looking at that top decile of scores, a large number of these frequent VOC mutations are captured. This is relevant when comparing to EVE alone, where we see much more were captured by EVEscape rather than just EVE. In fact, we can use this breakdown of EVEscape into its components by understanding whether a mutation might increase fitness or whether it might result in escape by looking at this comparison between EVE and EVEscape. For those mutations with high EVE score but low EVEscape, they might impact structure but do not escape. For instance, A222V results in an increased RBD opening, but is known to not reduce seroneutralization, while T547K stabilizes the down conformation of spike, which in turn enhances transmissivity. We can also look at how EVEscape and experiments like DMS can come together in order to capture frequent mutations. Here in the upper right-hand corner of this plot, we can see a significant number of frequent pandemic mutations would be captured by looking at EVEscape and experimental results together. We can also see, though, that EVEscape might be able to pick up interesting mutations that some experiments might have missed. So when looking at experiments and EVEscape together, one example of a mutation picked out by both is E484A. 484 has been identified by both, and it's also a site that is very mutable as well as accessible on the ACE2 binding region. On the right, we can see its interaction with the therapeutic antibody LICOV 555. This is a therapeutic antibody that lost its FDA authorization as a result of the spread of Omicron due to antibody escape. We can see its interaction with R50 and R96, and in Omicron, the mutation to an alanine results in a loss of these salt bridges, displacing the antibody. Therefore, we can see this as an example of understanding of fitness, understanding of accessibility, as well as the similarity comes together to result in kind of a real-world prediction that has been seen throughout the pandemic. Further escape mutations to Omicron are also identified by EVEscape alone. This includes mutations like S477N and T478K. Now we'd like to move beyond ranking single mutations early and instead ask the question, can we predict the next emerging VOC strain? So now later in a pandemic, looking at strain level information, could we actually predict what might next become dominant? So we can use EVEscape scores to actually aggregate across the combinations and mutations and look at the strain scores as a whole. VOC strains like BA4 have high predicted escape compared to common mutations. So we look here at comparison to random combinations and mutations at the same distance from Wuhan as that strain. And so we see that here BA4 has high predicted escape when compared to random combinations and mutations, but even when compared to frequent mutations, as well as mutations from other VOCs. We can then also look at how all of these strain level scores compare for frequent strains that have occurred throughout the pandemic. And we can see VOCs like Delta and Omicron stand out. As we continue to refine our model, as well as new strains emerge, we'll continue to update our predictions for these emerging strains and put them at evescape.org. So we have currently focused on just using pre-pandemic data. Surprisingly, pre-pandemic data has been able to capture escape mutations from both deep mutational scans, as well as frequent variants of concern mutations from the pandemic. We know now that there are pandemic-specific constraints that we might want to consider now later into the pandemic. And so now that we are this far into pandemic, we should think about what sorts of information we could include that could allow us to improve our model. Due to the modularity of the EVEscape framework, it's possible for different pandemic data to be integrated in different ways for each of the three components. For the fitness component, we could consider incorporating pandemic sequencing data into the training data for our fitness model that currently just uses deeper evolutionary sequences. Additionally, we could include results from the viral fitness DMSs that have information about expression and ACE2 binding. For accessibility, we could use knowledge of where known pandemic antibodies bind in order to focus EVEscape onto certain regions of spike. Thirdly, for dissimilarity, we could use known antibody interaction information as well as deep mutational scanning data to better characterize what mutations would displace antibody binding. We'd like to integrate EVEscape with models that learn directly from pandemic prevalence information. So an example of this is PIR0, which learns pandemic mutation effects from observed growth. This is in comparison to EVEscape, which uses deeper evolution to predict effects of previously unseen mutations. So while PIR0 learning directly from pandemic information might be good at getting granular differences between observed strains in order to predict what might become dominant in the immediate future, EVEscape is designed for the longer term, and so it might be useful for rare and unseen mutations, as well as mutations on new backgrounds. It then might be useful to bring these two together for a more medium-term anticipation of concerning strains. So while we can get the granularity from PIR0, we can also take advantage of understanding of unseen mutations that EVEscape allows. We can see EVEscape and PIR0 coming together here, where for seen mutations, as PIR0 identifies mutation effects from these seen mutations, we can see certain mutations being picked out as important by both of these models. So here we're looking at those mutations that have been seen by February of this year. We can see also the mutations that have been seen by September of this year. So while at first some mutations that have not been seen wouldn't have scores for PIR0, they would still have these early scores from EVEscape, where we can see on the upper right, new Omicron mutations could be predicted early with high EVEscape that are then later picked out by PIR0 as important as they've occurred throughout the pandemic. So this includes mutations like R346T and Y248N, which are two additional mutations on top BA2 that form BA2.76. In this direction, EVEscape can be used for pandemic preparedness going forward, as we start to think about incorporating pandemic data into EVEscape in order to anticipate new strains. We are in collaboration with Jeremy Lubin, Jacob Lemieux, and Mike Seaman, working to characterize emerging variants and forecast new escape strains. This is a project being funded by CEPI, which is the Coalition for Epidemic Preparedness Innovations, which seeks to develop a system for early warning of potentially concerning variants. So we are developing a system where EVEscape, using pandemic data, which is PIR0, can be used to both rank current strains and so to have an understanding of how impactful different emerging variants might be, as well as to anticipate new strains. We then prioritize some strains to design constructs and perform experimental validation, in this case, infectivity and neutralization assays. This will allow for a real-time early warning system that could identify potentially concerning patterns and mutations before they become dominant. I'd like to thank the members of the Marks Lab, OatML, as well as the CEPI collaboration. And it's now time to take any questions. Thank you very much.
Great, that was wonderful. Thank you so much. It's just very exciting to hear how quickly you were able to apply, real kind of fundamental science to the pandemic. So if anyone has any questions, please feel free to type in the Q&A or raise your hand. And you can actually just go ahead and unmute and ask them. I have a couple of questions. One was, do you think you would take this the next step? Would you take your most predicted impactful variants, actually engineer that virus and preemptively make sera of some sort? Would you think that would you see a place where that would be reasonable, or do you think that it's still better to just do really active surveillance and look to see for the real world experiment to take place?
Yeah, so I think we see this primarily for a pandemic as providing an initial reduction in where you need to look for vaccine development or for things like that. So I think we would see it primarily. I think it's difficult to say that we'd capture all the constraints that are relevant from the pandemic, at least with our initial model. We're hoping as we go forward to do better with that, incorporating more pandemic data. But I still think experimentation is really powerful, which is why the CEPI collaboration is really exciting for us. What we're kind of hoping to do is prioritize using our model and say, these are the mutations that we think are most likely to be concerning. And even these are the combination mutations that we think might occur together. So we can propose maybe a few variant strains that we think are plausible, and then quickly accelerate those into testing against sera samples. So our collaborators have great connections to get the most recent sera samples, not only from the original vaccines, but from people who've been infected with variants, as well as now that we have variant-specific vaccines. So we can quickly characterize our initial predictions against as close to a nice broad swath of sera as I think you can find anywhere, and then accelerate those into being used for vaccines and other things. So I think the experimentation side will always be important, but we see the role of computation as being able to really narrow down what you need to test, because these samples are also quite limited, right? One thing we've seen with this collaboration is they can only test a certain number of variants against these limited sera samples. And so we think it is really helpful to kind of prioritize what they look at.
That makes sense. And then is your model relevant for not just escape from immune response in terms of antibody production, but could you take it back and think of a model trained instead of on escape from non-human species, right? So if this came about from some sort of a non-human coronavirus, which obtained mutations that were then able to have high amounts of human infectivity, do you think an analogous data set that looked at perhaps the viral microbiome among species of a particular area, could you look at escape from those species, for example?
That'd be really interesting to think about. Yeah, I think it would be something that should translate to some degree, but I think we would have to carefully consider what the differences in immune constraints across those organisms are. I've actually been really curious about this recently. I've been trying to figure out what we know about bat immune systems. And the answer is not that much. We know a few things. So we know some things about the balances between B and T cells, and we know their VDJ recombination looks quite different than ours. And is actually probably more diverse than ours. And so they rely less on the somatic hypermutation side and more on their original repertoire diversity and things like that. So we could try thinking about how those things would affect a model and probably try to tweak those and see how we do. But yeah, I'm really curious to see as people start doing more of those experiments, especially in hosts like bats, which are super relevant for us, what we learn about the immune system there.
And just one more invitation, if anyone has any questions, feel free to type in the Q&A or raise your hand and we'll unmute you. And my last question then is whether your model has thought about incorporating kind of virulence or severity of illness predictions into it as well, because there's luckily been a decrease in hospitalization and mortality rates across the pandemic, which is multifactorial in its etiology. But escape escape from kind of antibody response may be inconsequential for some more mild mutations, or mutations that tend towards a more mild infection, whereas it could be hugely impactful if it also came along with, more severe microvascular injury or kind of other things. Is that something you feel you could incorporate in the model, or is just a separate consideration?
I think that is a really interesting thing to incorporate and something that we could think about. And this kind of plays into that broader question that I've heard a lot of chatter on, which is like, what is more advantageous to the virus in terms of virulence, right? And for a lot of the host organisms and the organisms we're training on more broadly, and bats in particular, right, their response to this virus is much less severe than ours generally, right? So they don't mount the same sort of inflammation response that we do. So it would almost be interesting to see if we could use the deeper evolution data in kind of a different way, right, to see where these new strains are different from what we've seen in deep evolution, and then see how that affects virulence, right? So yeah, I think that could be an interesting thing to incorporate, and it would be useful, especially at this stage in the pandemic, since we will care more about those strains. But I think it might require using the sequencing data in kind of a different way than we have so far and developing a different model on it.
Great. Well, thank you both for just a really exciting and so timely, relevant work that you shared with us. And so everyone can take a stretch, get more coffee, and then we'll reconvene at 9:30 for the main MPG session.
Great. Thank you so much for having us.
Thank you
Thanks
very
so much. Bye.